Hello everyone, I am Dr. Pooja Johari, your biochemistry educator. So today we are going to discuss a very important topic in biochemistry from lipid metabolism, which is ketogenesis. Now what do you understand by this? When it occurs, what is the clinical significance of ketogenesis? This is all what we are going to cover in today's video. So as the name suggests, ketogenesis, genesis means again synthesis. So how do we define this? It is simply synthesis of ketone bodies. Now what are these ketone bodies? Ketone bodies are one is acetone, second is acetoacetate, and third is Beta-Hydroxybutyrate. Right? So there are basically three ketone bodies: acetone, acetoacetate and Beta-Hydroxybutyrate. And the process of synthesis of these ketone bodies is ketogenesis. Now again, what all points we have to focus? As I have already mentioned that we should know the site where it occurs, what is the importance of this and in what conditions it increases. So if we talk about the site of ketogenesis, it occurs in the mitochondrial fraction of the liver. Right. And what is the precursor of ketone bodies? Precursor of ketone bodies is acetyl coenzyme A. And in what pathological conditions it increases? Mainly the production increases during starvation, and that is what we are going to discuss during starvation, how they increase, and during diabetes mellitus. So these are the two pathological conditions in which there is increased production of ketone bodies. Now, how the synthesis takes place? See, precursor is this acetyl coenzyme A. This acetyl coenzyme A combines with one more molecule of acetyl-CoA, forming acetoacetyl-CoA. And simply it is the synthesis, so we will say that synthase of acetoacetyl. Acetyl-CoA synthase. Now once this acetoacetyl-CoA is synthesized, it combines with one more molecule of acetyl-CoA to form HMG-CoA. Now this HMG-CoA stands for beta-hydroxy beta-methyl gluteryl-CoA. Beta-hydroxy beta-methyl gluteryl-CoA. Now since this is synthesis of HMG-CoA, so the enzyme is HMG-CoA synthase. Now this HMG-CoA undergoes splitting, and we all know that the splitting enzyme is lyase. Splitting of what? HMG-CoA. So the name of the enzyme will be simple HMG-CoA lyase. And this HMG-CoA, when it splits up, it gives rise to first ketone body, which is acetoacetate, giving out this CoA. So we can say that this acetoacetate is the primary ketone body or the first ketone body which is produced. Right? Now this acetoacetate will now undergo reduction in the presence of enzyme. Obviously the enzyme is dehydrogenase and forming the second ketone body or the secondary ketone body, which is beta-hydroxybutyrate. And if this acetoacetate undergoes decarboxylation, it gives out carbon dioxide forming acetone. Because you know acetone is volatile, which is produced as a result of decarboxylation. And the enzyme here is dehydrogenase and here it is decarboxylase. So now let's quickly see. First is this acetyl coenzyme A combines with one molecule of acetyl-CoA forming acetoacetyl-CoA, which again combines with this forming HMG-CoA. This HMG-CoA undergoes splitting in the presence of enzyme HMG-CoA lyase forming acetoacetate, which undergoes a dehydrogenation forming beta-hydroxybutyrate and decarboxylation forming acetone. So acetoacetate being the first ketone body which is synthesized, that is why it is known as a primary ketone body. Beta-hydroxybutyrate is the secondary and acetone is again secondary. So this is what is ketogenesis, which is taking place where? It is taking place in the mitochondrial fraction of the liver. Right? Now, the normal concentration of ketone bodies is 1 milligram per dl. Right? So, if we talk about this, it is also, you can say, excreted in urine, but it is insignificant. So, this is the normal concentration. When the concentration of ketone bodies increases in the blood, it is known as ketonemia. If it appears in urine, it is known as ketonuria. And when it is associated, means there is increased concentration of ketone bodies related to diabetes, it is known as diabetic ketoacidosis. Right. Now see, always remember whenever we talk about metabolism, this is the anabolic part. Whenever ketone bodies have to give energy, they have to break. So breakdown of ketone bodies will be known as ketolysis. So ketogenesis is synthesis of ketone bodies, ketolysis is breakdown of ketone bodies. Now see, synthesis of ketone bodies always takes place in the liver, but utilization, this is very, very important, utilization of ketone bodies is never by the liver. Now, what is the reason behind that? If we talk about this, because liver lacks thiophorase enzyme. That is why utilization is by extrahepatic tissues. And if we look at ketolysis, this is how it occurs. This acetoacetate will be converted into acetoacetyl-CoA. Now see, if we look at the terminology only, what is the basic difference between acetoacetyl-CoA and acetoacetate? It is the difference of this CoA, plain and simple. So, and this CoA is coming from succinyl coenzyme A. What is happening is, this succinyl coenzyme A, it is giving this coenzyme A to acetoacetate and it is converted back to succinic acid. Here the enzyme is thiophorase, and this thiophorase enzyme is absent in the liver. That is why utilization of ketone bodies is never by the liver, even though production of ketone bodies is there in the liver. Right? So this is a very, very important point. The liver lacks thiophorase, utilization is by extrahepatic tissues. Then the most important point about this ketogenesis or ketone bodies is the test to detect the presence of ketone bodies, because ketone bodies are the most important constituents, or you can say important abnormal constituents of urine. Now, how they are important constituents of urine? Because I told you the normal concentration is 1 milligram per dl, even which is excreted in urine, but it is insignificant. And increased concentration of ketone bodies is observed during starvation and during diabetes mellitus, because what happens is, whether we talk about starvation or whether we talk about DM, what is the situation of the body like? Because in diabetes also there is increased concentration of glucose, but it is not being properly utilized by the organs. So what happens is, they have to survive on some other mode, and that some other instantaneous source of energy is the fats. And these fats, they form acetyl coenzyme A. So we can say that increased during starvation and DM, there is increased concentration of this acetyl coenzyme A, which will be diverted to form these ketone bodies. And these ketone bodies are detected by a very, very important test, which is known as Rothra's test. Rothra's test is the test to identify the presence of ketone bodies in urine, and this test is given only by acetoacetate and not by Beta-Hydroxybutyrate. Right, now how do we carry out this Rothra's test? What happens is, first of all, we saturate the sample of urine. saturate this urine sample with ammonium sulfate. Right? Then after that we add sodium nitroprusside, and after addition of sodium nitroprusside we add liquor ammonia along the sides of the test tube. Liquor ammonia along the sides. Of the test tube. So there is formation of a violetish ring at the junction. So we say that it gives positive Rothras test. So always remember that positive Rothras test will be there in the sample of urine of the patient suffering from diabetes mellitus and during starvation also. One more test is there which is known as Gerhard's test. In Gerhard's test also again it is given only by acetoacetate. And not by beta-hydroxybutyrate. Right? So this is what is ketogenesis and ketolysis. Main thing is what you have to remember in this, that it takes place. First of all, how do we define it? It is simply the process of synthesis of ketone bodies. Acetone, acetoacetate and beta-hydroxybutyrate. If you see, acetoacetate is an acid, beta-hydroxybutyrate, it is again an acid. होता कहाँ पे? Where it occurs? It occurs in the mitochondrial fraction of the liver. This you have to remember, that always remember that it is ketone bodies are synthesized in the liver in the mitochondria. Precursor is acetyl coenzyme A. This acetyl coenzyme A combines with one molecule of acetyl coenzyme A forming acetoacetyl-CoA, which combines with one more molecule of acetyl coenzyme A forming HMG-CoA, which stands for beta-hydroxy-beta-methyl-gluteryl-CoA, which undergoes splitting. So this HMG-CoA is the most important. In fact, this is the deciding enzyme of ketogenesis, which forms the first ketone body, which is acetoacetate. And this acetoacetate undergoes dehydrogenation forming the secondary ketone body, beta-hydroxybutyrate and acetone. Acetone is volatile, so it will be evaporated as such. Then this is the normal concentration of ketone bodies. When it increases in blood, it is known as ketonemia. When it increases, it appears in urine, it is known as ketonuria, and when it is associated with diabetes, it is known as diabetic ketoacidosis. What happens here is ketoacidosis means there is high acidic environment in the body because of acetoacetate and beta-hydroxybutyrate. So the pH of the body also drops down. Then in a normal individual also ketone bodies are produced, but ketone bodies play a very important role in giving energy. So breakdown of ketone bodies is ketolysis. Now a very important point to remember in this is, even though the synthesis takes place in the liver, liver never ever utilizes ketone bodies as the energy fuel. Why? Because liver lack thiophorase enzyme, which is actually responsible for converting acetoacetate into acetoacetyl coenzyme A. So this is actually a irony or tragedy with the liver, even though synthesis takes place but utilization never takes place. Now the test required, the test for the detection of ketone bodies, most important from examination point of view, it is a Rothras test. Another test is there, Gerhardt's, but the Rothras test is carried out. What we do is, it is answered only by acetoacetate. That this is also a question asked in the exam, that which of the following ketone body does not answer Rothras test. So always remember, it is never answered by beta-hydroxybutyrate. It is always answered by acetoacetate. Acetone it comes out in the form of that effervescence. So what we do is, we saturate the sample of urine with ammonium sulfate, then we add sodium nitroprusside, and then we add liquor ammonia along the sides of the test tube. So formation of that violetish ring at the junction detects the presence of ketone bodies, so it is positive Rothras test. Another is Gerhardt's test, which is only again answered by acetoacetate and not by beta-hydroxybutyrate. So this is what is ketogenesis and ketolysis. So that's it about ketogenesis. Thank you so much.